Usually when we talk about a medical diagnosis, there's this expectation of like mechanical precision.
Right.
You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is, broken or not broken, it's clean.
Oh, totally.
But when you step into the world of microbiomes, and specifically the human vaginal microbiome, that x-ray machine just shatters. We're suddenly looking at a landscape that is incredibly murky, highly volatile, and honestly deeply misunderstood.
It is the absolute definition of diagnostic muddy waters. I mean, considering this ecosystem affects literally half the global population, it is rarely discussed with the detail or even the nuance that it actually demands.
Which is why we are taking you on this custom-tailored deep dive today. We have a towering stack of sources in front of us, ranging from clinical reviews on the genitourinary syndrome of menopause, or GSM, to some really cutting-edge genomic research and evolutionary biology.
Yeah, the data in here is incredible.
It really is. And our mission today is to cut through that murky water and answer one very specific, very crucial question. Why does the loss of a simple sugar called glycogen, specifically in aging epithelial cells, lead to the total collapse of an entire protective bacterial ecosystem?
Well, to even begin answering that, we really have to throw out a lot of what we, assume about human biology.
So for sure.
Because in almost every other part of the human body, like your gut, for example, high microbial diversity is the hallmark of health.
Right. You want the variety.
Exactly. You want a massive, thriving variety of different bacterial species. But the ideal human vaginal microbiome completely flips that script. It is surprisingly undiverse.
It's weird, right?
It's essentially a monoculture. It's ruled by a single dominant genus of bacteria, which is Lactobacillus.
It's a monoculture, which goes against literally everything we hear in those probiotic commercials where diversity is good.
Right. But here, a monoculture is the absolute goal. So, to understand why losing a simple sugar causes a total collapse of this unique ecosystem, we first need to figure out why this monoculture is here in the first place.
Yes, we do.
And doing that means zooming way, way out. We have to go back to human evolution.
Looking at the comparative data across mammalian species in our sources, there's just a staggering evolutionary divergence. Humans are entirely unique here.
Okay.
In healthy human females, as we just established, lactobacilli make up more than 70% of the vaginal microbiome. But if you look at other mammals like dogs, cats, primates, that number is incredibly low.
How low are we talking? Like 10%?
Less than 1%.
Less than 1%. Wait, really?
Yeah.
That isn't just a slight variation. That is a massive biological chasm.
It's a completely different ecological strategy, honestly. I mean, in other mammals, the vaginal pH does drop slightly during periods of high estrogen, but it never, ever reaches the extreme protective acidity that we see in humans.
Right. The median vaginal pH in humans is around 4.5. In other mammals, it ranges anywhere from like 5.4 to 7.8, which is far more alkaline, much closer to neutral.
So what happened? Why did human biology suddenly decide, it needed a highly acidic, lactobacillus-dominated monoculture? Our sources point to a really compelling idea called the starch hypothesis.
The premise here is that human biology fundamentally shifted alongside human agriculture.
Right, when we started farming.
Exactly. As our ancestors moved away from strictly hunter-gatherer diets and began consuming high levels of agricultural starch, think grains, tubers, early crops, our bodies had to figure out what to do with all that extra carbohydrate energy.
Yeah, that makes sense.
The data suggests this dietary shift actually led to increased levels of glycogen being deposited directly into the tissues of the human vaginal tract.
Okay, let's unpack this. So the vaginal lining is essentially a self-stocking pantry.
I love that analogy, yeah.
It's a wall of cells that plump up with glycogen, which is just stored sugar thanks to the presence of estrogen. And as those surface cells naturally shed and break open over time, they release a constant food supply for the good bacteria.
That is a brilliant way to visualize it. It is a self-stocking pantry. But the host isn't just throwing raw ingredients at the bacteria and hoping for the best.
What do you mean?
The biochemistry of how this pantry operates is incredibly sophisticated. The host is actually actively prepping the meals.
Oh, wow.
Yeah. Human alpha amylase enzymes, which are naturally present in the vaginal fluid, act like little biochemical scissors.
Wait, alpha amylase, isn't that the same enzyme we have in our saliva?
It is, yeah.
The one that makes a plain cracker taste sweet if you chew it long enough because it's breaking down the starch.
The very same mechanism. The alpha amylase in the vaginal fluid clips this massive, complex glycogen molecule into bite-sized fragments called oligosaccharides.
So it's basically doing meal prep.
Exactly. The host is doing the heavy lifting, chemically speaking, so the lactobacilli can easily feast on these pre-cut sugars.
And nothing is free in nature. So what does the human host get in return for running this biochemical soup kitchen?
Well, in return for this perfectly prepped meal, the lactobacilli produce lactic acid, and they pump out massive amounts of it, reaching levels of 100 to 150 millimolar.
That sounds like a lot.
It is. This specific byproduct is what drives that incredibly low pH, securely anchoring the environment between 3.5 and 4.5. It basically creates an acid mantle that makes it incredibly hostile for foreign pathogens to even survive.
An invisible chemical shield.
Exactly.
But there was a detail in the genomic research about this lactic acid that forced me to, reread the page twice.
Oh, the histones.
Yes. It isn't just a passive shield sitting on the surface. It is actively interacting with the human host's DNA.
This is where the symbiotic relationship becomes truly profound.
Yeah.
The specific type of acid-produced lactic acid has the ability to block something called histone diacetylases inside the human host cells.
Okay, for those of us who haven't taken cellular biology in a few decades, what does blocking a histone deacetylase actually accomplish?
So think of histones as little spools that your DNA wraps around.
Go.
If the DNA is tightly wound up on the spool, the cell can't read the genetic instructions to make repairs. By blocking the enzyme that tightens those spools, the bacterial acid essentially forces the human DNA to stay unspooled and open.
No way.
Yeah. This actively enhances gene transcription and DNA repair within the human epithelial cells.
That is mind-blowing. The bacteria are actively hacking the host's cellular machinery to help maintain and repair the health of the very pantry they live in.
Yay.
They are fixing the walls of their own house.
It is an evolutionary masterpiece of cooperation.
Right.
But it has a critical vulnerability.
The estrogen.
Exactly. This entire feedback loop relies on estrogen to signal the initial stocking of that pantry with glycogen.
Which brings us to the core mystery we set out to solve today. If estrogen is the keymaster, turning the lock to supply the glycogen, what happens when that hormonal supply chain shuts down during menopause?
Well, we see a cascading physiological process known as atrophy. As systemic estrogen drops, the vaginal epithelium, that thick wall of cells that makes up our pantry, stops receiving the signal to plump up and multiply.
Okay.
So the wall begins to thin out significantly.
And if the wall is thinner, the natural shedding process has to change, right?
Drastically. Fewer of those superficial squamous cells are sloughed off into the vaginal lumen. The physical delivery of food just slows down.
Oh, I see.
And worse, the cells that are shed have a severely depleted glycogen content because the estrogen isn't there to pack them full of sugar in the first place.
So the food supply completely dries up, the lactobacilli literally starve,
Yes.
their populations crash, the lactic acid production halts, and without that acid, the pH begins to creep up past 5.0.
Exactly. It's a total ecosystem collapse.
So what does this all mean? I want to push back on this a little. If the issue is simply a lack of sugar, why wouldn't the ecosystem just pause?
What do you mean?
Well, if I stop putting birdseed in my yard, the birds just fly away and the yard gets quiet. It's empty. Why does a lack of glycogen actively lead to severe infections and the painful symptoms of genitourinary syndrome of menopause? Why doesn't it just become a neutral, inactive zone?
Because microscopic nature abhors a vacuum. An ecosystem in the human body never just pauses. It gets aggressively colonized by whatever organism is best equipped to survive the new conditions.
Oh.
When that pH was hovering at 4.5, it acted like a chemical force field, keeping opportunistic, bad bacteria in a state of suspended animation. They were there, but they were completely dormant.
So the acid was keeping them asleep.
Basically. But once the lactobacilli starve and the pH rises above 5.0, that force field drops. Those dormant bacteria, specifically strains like Gardnerella, Prevotella, and Streptococcus, they sense the lack of acid and immediately wake up.
The bad actors seize the territory. And this shift is what drives the clinical symptoms of GSM, which our sources note affects an astounding 50 to 70% of postmenopausal women.
It's a huge number.
We are talking about chronic dryness, intense itching, dyspareunia, which is severe pain during sex, and a relentless cycle of recurrent urinary tract infections.
And this is a vital distinction to make. These symptoms aren't just a vague, inevitable consequence of, getting older.
Right.
They are the direct mechanical result of a specialized ecosystem collapsing, allowing dysbiotic bacteria to launch a hostile takeover.
Okay, but if it's a hostile takeover and
Mhm.
the estrogen has stopped delivering the main food source, what are these invaders even eating?
That's the million-dollar question.
If it's a literal battle for food in a starving environment, how do the good bacteria try to feed themselves compared to the bad ones? Are they just fighting over the same scraps?
Actually, no, they aren't fighting over the same scraps because they have completely different biological weapons, specifically their enzymes.
Okay, break that down for us.
If we look at the genomic data, let's take one of the primary protective strains, Lactobacillus crispus. It utilizes a very specific genetic tool to eat. It possesses a gene called PULE, which produces an enzyme known as a type I pullulinase.
Lulunase. Uh, what does that specific enzyme do?
So glycogen is a highly branched molecule. Imagine a thick, gnarly oak tree.
Okay, visualizing a tree.
This pullulinase enzyme is designed to target very specific points on that tree called alpha-1, 1, and 6 branches. Lactobacillus uses this enzyme to carefully snip those specific branches, importing the nutrients directly into its cell to feed itself. It's a very targeted, very polite way of eating.
Okay, but then you have the invaders. The genomic sources highlight specific Gardnerella strains, Gardnerella vaginalis, Huzinski, and Leopoldi. I'm guessing they don't have this polite little pullulinase enzyme.
Not at all. These dysbiotic bacteria possess incredibly aggressive alpha-glucosidase activity.
Oh boy.
Yeah. While Lactobacillus is carefully snipping specific, easy-to-reach branches, Gardnerella is capable of degrading raw, ungelatinized amylose.
Ungelatinized amylose. That sounds dense.
It is an incredibly tough, complex alpha 1 over 4 glucan molecule. It's a form of carbohydrate that the polite Lactobacillus simply does not have the tools to break down.
It sounds like Lactobacillus is a neat eater, drinking from a juice box like it imports the specific nutrients directly into its cell without leaving a mess.
Yes.
But Gardnerella is like a mob smashing a piñata. It uses brute force to shatter these tough, complex carbs, and in the process, it leaves sugary crumbs scattered everywhere.
What's fascinating here is that researchers can actually see those metaphorical piñata crumbs in the clinical data.
Really?
Yeah. When scientists measure the different types of sugars present in vaginal fluid, the measurements perfectly validate that analogy.
What do those measurements actually look like?
Studies show that high levels of intact, complex glycogen strongly correlate with high lactobacillus populations and a healthy, low pH.
Makes sense.
But high levels of maltodextrin, which is the scientific term for those breakdown crumbs you mentioned, correlate almost exclusively with a dysbiotic takeover.
Because the Gardnerella smashed the piñata.
Precisely. Gardnerella aggressively fractures the remaining complex carbohydrates in the environment, releasing abundant maltodextrins. And this is where their strategy becomes truly devious.
Oh.
Those maltodextrin crumbs feed the entire community of bad bacteria.
Oh, I see. They aren't just feeding themselves. They are throwing a feast for the rest of the pathogenic army.
They supply the broader dysbiotic community with a highly accessible, easy-to-eat carbohydrate nutrient pool. The starving Lactobacilli, with their neat-eating juice box strategy, are just completely outcompeted by this aggressive, cooperative feeding frenzy.
That is just wild to visualize.
Yeah.
The Lactobacilli are starved out because the estrogen stopped delivering the easy food,
Mm-hmm.
and then the invaders break down the tough scraps left behind to feed a growing army.
It really is microscopic ecological warfare.
So for you listening, how do we actually apply this knowledge? When dealing with the severe realities of GSM, how can we step into this microscopic war zone and help restore order?
Well, the clinical interventions detailed in our sources generally divide into two main approaches, hormonal and non-hormonal. Because the root cause of the collapse is the loss of estrogen driving the glycogen supply, hormonal options are incredibly effective at fixing the problem at its source.
You basically just restart the supply chain.
Exactly. Low-dose vaginal estrogen is a primary, highly localized treatment. It signals the epithelium to plump back up, restarting the shedding of glycogen-rich cells, which feeds the lactobacilli and restores the acid mantle.
That's great.
There is also vaginal DHEA, which the body converts locally into active hormones, and an oral medication called ospemifene. That one specifically mimics estrogen in the vaginal tissues to reverse the atrophy without acting as an estrogen everywhere else in the body.
But there is a massive population for whom hormones simply are not an option. Breast cancer survivors or those with a high risk of estrogen receptor-positive cancers often cannot or choose not to use hormonal treatments, even localized ones.
That's very true.
For them, over-the-counter non-hormonal lubricants and moisturizers are the absolute first line of defense to manage the pain and dryness.
And providing effective symptom management for that population without introducing systemic risk is a critical aspect of clinical care.
Right. But if someone is dealing with severe dryness and tissue damage, my instinct would be to just grab the heaviest, most intensely moisturizing product on the shelf.
Right.
Like more moisture, thicker product equals better relief, right?
That is a very common instinct, and unfortunately, it is the exact instinct that can lead to further tissue destruction.
Really?
Yeah, you cannot just introduce random chemistry into an ecosystem this fragile.
Here's where it gets really interesting. Are you saying an over-the-counter moisturizer could actually make the bacterial dysbiosis worse?
It absolutely can, and frequently does. The sources dive deep into the specific chemical metrics consumers must look for. First and foremost, a product must be pH balanced to the specific environment.
Meaning it needs to match that 3.5 to a 4.5 acidic range to support the lactobacillus force field. Because if you use an alkaline product, you're just rolling out the red carpet for the Gardnerella.
You'd be actively suppressing the good bacteria and helping the pathogens wake up. But the second metric is just as critical and almost completely ignored in mainstream marketing, which is osmolality.
Osmolality. Okay, let's break that down because I rarely see that word on a pharmacy box.
Osmolality refers to the concentration of dissolved particles like salts, sugars, and preservatives within a liquid. For a product to be safe for this environment, it must be isoosmolar.
Isoosmolar.
Nate, that means the concentration of particles in the moisturizer must exactly match the natural concentration of the body's own cells.
What are the actual numbers we need to look for to ensure it matches?
The ideal osmolality range is between 280 and 310 millismoles per kilogram. The absolute strict upper limit recommended by health organizations is 1,200.
And what happens if someone unknowingly buys a product off the shelves and it's hyperosmolar? Let's say it's packed with heavy glycerin or thickeners and it's way over 1,200.
If a product is hyperosmolar, it has a much higher concentration of particles than the cells of the vaginal lining. And through the basic laws of osmosis, water will always move toward the higher concentration of particles.
Oh, no.
Yeah. So that moisturizing product will literally suck water out of the already thinning epithelial cell.
Oh, that is a cruel irony.
Yeah.
You're using a product designed to moisturize, and it actively dehydrates you at a cellular level.
It pulls the water out, causing severe cellular dehydration. Those fragile cells shrivel, which leads to microscopic tearing and microtrauma. You are basically exacerbating the exact tissue damage that is causing the pain and further destroying the environment the lactobacilli need to survive.
That is incredibly practical, actionable knowledge. The pH and the osmolality aren't just like marketing buzzwords to ignore.
Not at all.
They are the literal physical parameters required to keep this microscopic ecosystem from collapsing further.
They dictate whether a product acts as a supportive scaffolding for the mucosa or a wrecking ball.
Wow. Let's take a moment to look back at the incredible journey we've just been on today. We started with an evolutionary quirk, the starch hypothesis, showing how early human agriculture fundamentally shifted our biology, creating a unique, glycogen-fueled, highly acidic microbiome completely unlike any other mammal on Earth.
We saw how estrogen acts as the crucial signal, keeping our cellular pantry stocked, and how the bacteria use that food to produce acid that literally helps repair our own DNA.
It's still blowing my mind. And then we explored how the loss of that estrogen during menopause stops the shedding of those cells, literally starving our protective lactobacilli,
Right.
which drops the chemical force field and allows aggressive bacteria like Gardnerella to smash the piñata, feast on the maltodextrin crumbs, and trigger all the painful disruptive symptoms of GSM.
And we clarified that managing this collapse requires precise chemistry, whether through restoring the hormonal supply chain or carefully managing the environment with pH-balanced, strictly iso-osmolar products.
For you listening, understanding the why behind genitourinary syndrome of menopause does something incredibly important. It completely removes the stigma.
Yes,
This
absolutely.
isn't a personal failing, it isn't a hygienic issue, and it isn't just a vague, untreatable side effect of aging. It is a fascinating mechanical, ecological shift. It is a biological chain reaction, and once you understand the links in that chain, you can treat it.
If we connect all of this to the bigger picture, there is one final unexplored thread from our sources that I think is truly profound to consider as we wrap up today.
Lay it on us.
We've established today that estrogen and diet drive this ecosystem, but what about our actual DNA?
Okay, where are we going with this?
Researchers are now looking deeply at host genetic modifiers. Specifically, they're studying genes like GYS1, which controls how our bodies synthesize glycogen in the first place, and the SLC2A1 gene, which controls glucose transport across our cell membranes.
Meaning natural variations in how different bodies handle sugar on a deeply genetic level.
Exactly. This raises an incredible possibility for you to ponder long after this deep dive ends. Could your innate genetic ability to synthesize and transport sugars dictate the resilience of your microbiome before you even factor in hormones or diet or menopause?
Wow.
Are some people genetically hardwired from birth to have a perfectly stocked cellular pantry, while others are biologically predisposed to struggle when the microbial turf war begins?
That is a staggering thought to leave on. The idea that our resilience to these microscopic battles might be written into our genetic code before the fighting even begins. We want to thank you so much for joining us on this deep dive. Keep questioning the things we take for granted, keep exploring the hidden biological worlds inside us, and we will catch you next time.